Currently, induced pluripotent stem cells, or iPS cells, are mainly produced by hand, facing the problems of limited cell productivity and high production costs. To overcome these problems, the MyIPS project has developed a procedure for automated production of iPS cells using a specific instrument for a closed, automated cell processing system. Blood from a donor is first transferred to the instrument's chamber to isolate and collect the peripheral blood mononuclear cells, or PBMCs. To generate iPS cells, PBMCs and Sendai virus carrying the Yamanaka factors are added to the culture chamber to infect the cells. Following an optimized protocol of supplementing and changing the culture medium in this closed system, iPS cell colonies are formed and can be observed with the attached camera. iPS cells are collected after approximately 3 weeks of culturing. The collected iPS cells can be frozen for future use or transferred to the instrument for induced differentiation. Specific target cells can be differentiated in the instrument's chamber by providing the appropriate medium. Once the target cells have been obtained, they can then be used for therapeutic purposes.